So last time we finished off talking about the very first time you encounter an antigen, and that's your primary response. So the first time you encounter an antigen, it takes your immune system approximately 7 to 10 days to make antibodies. And remember, the first type of antibody produced are those big IgM antibodies, those pentamers, and then later your immune system switches over to IgGs. Now, because you have memory cells, memory B cells, memory T cells, the next time you encounter that exact same antigen, you're going to be able to produce those antibodies much faster in as little as 24 hours. So what that means is you have long-term protection against that particular pathogen. Hey, Roxy, check your settings. You might have to refresh in order to get your microphone to work. But everyone else can hear me? Yes, so. So every subsequent exposure, you're able to make those antibodies so fast that you don't get infected and succumb to that infection again. But as we've discussed, bacteria and viruses mutate and change. So it's very likely that pathogen is mutated just enough that its antigens are different enough, all the antibodies you have are no longer effective. So you have to get sick all over again, make more antibodies in 7 to 10 days, and recover. But the idea is that because of those memory cells, you will be able to respond much more quickly. And the type of antibody produced upon all subsequent exposures are IgG antibodies. Now, we talked about how the most antigenic substances are proteins and polysaccharides. Well, your body is full of proteins and polysaccharides. So why doesn't your immune system attack your body? And the reason why is that during fetal development, the self-recognizing T and B cells are selectively destroyed. And they used to believe that all of the self-recognizing T and B cells were destroyed. In fact, not all of them are destroyed, at least in some people. But those self-recognizing T and B cells are kept suppressed by the regulatory T cells. But if the self-recognizing T and B cells become unsuppressed, if they're reactivated, then they can start to attack. And that can lead to autoimmune diseases, which we're going to talk about later. So once again, those all-important antibodies, right? Antibodies are very specific for the antigens that they bind to. Remember that some antigens are huge, so that there may be multiple regions on those antigens that can be recognized by different antibodies. And we call those different regions epitopes or antigenic determinants. But wherever an antibody binds, that is a signal to the immune system to attack. And the problem, as we're going to see when we talk about hypersensitivity reactions, if those antibodies bind to your cells and tissues, your immune system will attack your cells and tissues. All right, so antibodies are just a signal that tells the immune system, come and attack. So we've already talked about helper T cells and how important they are, right? So they are important for regulating the overall immune response. And I remember that a diagram of the central role of the helper T cell. So helper T cells do help to regulate the overall immune response. They stimulate B cells and cytotoxic T cells. So just to refresh your memories, some kind of antigen-presenting cell like a macrophage or dendritic cell will engulf and digest an antigen and present pieces of that antigen on its surface along with the major histocompatibility complex. Then that antigen-presenting cell takes what it's found to a helper T cell. In the process, the antigen-presenting cell will secrete interleukin-1, which stimulates a helper T cell. Helper T cells secrete interleukin-2, which feeds back on the helper T cell to stimulate it even more. And interleukin-2 also helps stimulate cytotoxic T cells and B cells. So cytotoxic T cells, as the name tells you, are toxic to cells. And so cytotoxic T cells actually reach out, touch, secrete these toxic chemicals, and kill other cells. And the types of cells they kill are things like cancer cells or cells infected with viruses. Many of your cells are actually very altruistic. They will sacrifice themselves. And if they're infected with a virus, they send out a signal to the immune system to say, hey guys, I'm infected, come kill me. So that's one way that your immune system can eliminate the viruses, is by eliminating your cells that are infected with those viruses. Now, cell death technically is called apoptosis, although it's spelled apoptosis, and some people do say apoptosis, but that second P is actually silent. So it's apoptosis, and that's cell death. Regulatory T cells used to be called suppressor T cells because that's what they did. That's one of their main functions, is to suppress other cells. And regulatory T cells do help to suppress those self-recognizing T and B cells that could potentially attack you. But they also help to tell the immune system when to stop fighting. Remember, we talked about how this is all-out war. If a pathogen gets through your first and second lines of defense, you have this last line, your adaptive acquired immune response, which is very specific to that pathogen. But your immune system is going to use everything it has, right, to attack that pathogen. T cells, B cells, complement, everything, right, to eliminate that pathogen. But how does your immune system know when the war is over? Well, that's one of the things that regulatory T cells do, is to tell the immune system, okay, guys, you can stop fighting, we won. So they're very important T cells. Now, there are other types of T cells and subpopulations of T cells we're not going to go into because we don't have time for that. But remember, we're covering all the key components, right? The major players in the immune response. Now, when I talked about the different mechanisms of antibody action, I did mention the antibody-dependent cell-mediated cytotoxicity, or the ADCC. So this is used when trying to eliminate large parasites like worms, right? Because these are big organisms. And so what happens is antibodies cover that parasite, and that attracts immune cells like natural killer cells to attack that larger parasite. So natural killer cells are a population of immune cells. They're not T cells, but they're a different population of immune cells, and their job is to go around and kill things. So again, another piece to the puzzle of many different cell types that you have as part of the immune system. When it comes to active versus passive immunity, active means you're actually making the antibodies, whereas passive means you're receiving those antibodies. The benefit of active immunity is you have those memory cells, so you get long-term protection. It's considered natural active immunity if you make antibodies in response to an infection. It's considered artificial active immunity if you're making the antibodies in response to a vaccine. But you're still making the antibodies, so that's called active immunity. But vaccines are considered an artificial way to produce active immunity. We talked about how vaccines could be made of pieces of pathogens or maybe dead pathogens or inactivated toxins like the tetanus vaccine or diphtheria vaccine. Those vaccines are used to generate antibodies against those toxins, not the pathogen itself, but the toxins secreted by those pathogens. Because your immune system is perfectly capable of eliminating the pathogen, but that takes 7 to 10 days. You'll be dead from the toxin by then. So by immunizing you against the toxin, you can neutralize the toxin with antibodies and give your immune system time to kill those pathogens. So that's active immunity. When it comes to passive immunity, you're receiving foreign antibodies, so you're not making them. You have no memory cells. So passive immunity is very short term. It's considered natural passive immunity, as in the case of mom and her antibodies. Remember, IgG antibodies can cross the placenta. So all of mom's previous immunity to things she's been exposed to, her IgG antibodies can cross the placenta and protect that developing fetus. When the baby is born, mom's first milk, which is called colostrum, is very rich in antibodies and antiviral compounds. So the very first milk is really important immunologically for that newborn. But it's passive, right? Because all the antibodies came from mom. It's considered artificial passive immunity if you're receiving antibodies from another animal. So not a human, but another animal like a horse. And so we talked about this before with things like antivenoms, where they make antibodies against snake venoms, spider venoms. And the idea here is that if you get bitten by a poisonous snake or spider or something, they can inject you with antibodies which will neutralize the venom so that it's not toxic to you. But remember, these are foreign antibodies. So because they're foreign, your immune system is going to eventually destroy them. But not immediately, right? Because it takes 7 to 10 days. But your immune system may have become sensitized, and we'll talk about sensitization in the next lecture today. And that the next time you receive the antivenom, your immune system may overreact to it. And that can lead to host damage and death. And we're going to get into that. But that's active versus passive immunity. Any questions on that? Okay, next we're going to talk about immune disorders and hypersensitivity reactions. So we've seen how the immune system normally functions, and normally it functions extremely well, but sometimes the immune system misbehaves. Sometimes it overreacts to things that it should not be reacting to at all. And in the process, the hosts may be damaged or killed. So we call these kinds of reactions hypersensitivity reactions. And these are overreactions by the immune system. We're also going to talk about allergies, transplants, and autoimmune diseases. So autoimmunity. But all these things that we're talking about are things that we do not want the immune system to do. So when it comes to hypersensitivity reactions, the offending antigen is referred to as an allergen, but an allergen is just an antigen that causes a hypersensitivity reaction. And the problem is that the host is often damaged and sometimes dies as a result. But normally you do have to be sensitized to the allergen first. And there are 4 or 5 types of hypersensitivity reactions. Here in the US, we typically just use 4 types. In Great Britain, they have a 5th type, but as we'll see, that's what we consider a type 2 reaction. But the first type of hypersensitivity reactions, type 1, are anaphylactic reactions. These reactions are mediated by the IgE antibodies. Remember, we talked about IgEs, and their important function is to attack larger parasites, but unfortunately, they're involved in the allergic response. So the IgEs are attached to mast cells, and we see the IgE antibodies, and again, they act like a receptor or an antenna. If the allergen binds to the IgG, it causes the mast cells, which are full of histamines, to release those histamines. And histamines are one of our chemical mediators. And remember, chemical mediators help to stimulate the immune response, but a little bit goes a long way. You don't need too many chemical mediators released. If too much of any given mediator gets released, that can help lead to an overreaction by the immune system. And that's what happens in type 1 reactions. So the histamines get released. They cause symptoms like itching, edema or swelling, vascular dilation. And this can also cause some smooth muscles to contract, like in your respiratory tract. So there is some bronchial constriction, but it's not technically asthma, because asthma requires additional chemical mediators to be produced. So in order to get severe bronchial constriction that's characteristic of asthma, you also need the release of leukotrienes and prostaglandins along with histamines in order to cause that reaction. But it's the histamines that do cause the symptoms of allergies. And the location of the symptoms generally depends on where the mast cells are and where the histamines get released. In the respiratory tract, we talk about the typical symptoms of hay fever. So if you're allergic to tree pollen or grass pollen, then you suffer these typical symptoms, especially in the spring. Itchy, watery eyes, runny noses, sneezing, right? Because the histamines are being released in your nose, right, in your sinuses, and that's why you get those symptoms. But tree pollen and grass pollen are not harmful, right? They're not dangerous. Your immune system should be ignoring them, but in some people, they become sensitized, right, to these environmental pollens and things like that. So their immune systems are overreacting. When it comes to the intestinal tract, food allergies typically do not manifest themselves in the gastrointestinal tract. You typically get things like hives. Hives are these raised red itchy bumps or patches you get on your skin, and so they're caused by an allergy to something. And hives are sort of strange because they could occur in a very limited area of your body, or they could occur all over. And they can come and go. You could be covered with hives, and by the time you get to the doctor, they're all gone, right? They disappeared. And then when you leave the doctor, they all reappear. So hives can come and go. They could last for days, even weeks, and they're very, very itchy. Now, systemic anaphylaxis is very, very serious, and we are going to talk about that in more detail. So you could eat something, have a systemic anaphylactic reaction, and die. And we're going to go into that shortly. But many people often confuse a food intolerance with a food allergy, and they are not the same thing. A food allergy, you're actually making an immune response, like making antibodies. A food intolerance has nothing to do with the immune system. And the sort of classic example is lactose intolerance, right? So lactose, that sugar that comes from milk and dairy products. So human beings have the enzyme lactase. Lactase can break down lactose into glucose and galactose. But milk is something that is used in the animal kingdom, right, to feed newborns, right, to feed the young. And when it comes to humans, we typically only breastfeed for a few months, generally 3 months, maybe longer, but an average of 3 months. And then we don't breastfeed anymore. And, as humans evolved, that was the last time humans had milk. So shortly after breastfeeding stops, human beings stop producing lactase. And thereafter, they become lactose intolerant because once the genes for lactase are turned off, they typically stay turned off. Now, when it comes to dairy products and Europeans, about 10,000 years ago, when Europeans settled down and started farming and raising animals, they began consuming a lot more milk and dairy products like cheese. So in the period of about 10,000 years, Europeans evolved to constantly produce lactase. So most Europeans produce lactase constitutively now, right? Because dairy became a major part of their diet. But the rest of the world, that didn't happen, right? Because most of the rest of the world, they didn't, consume milk or cheese and that stuff. So that's why most of Asia and Africa, the people there do tend to be the most lactose intolerant. But Europeans relatively quickly evolved the ability to constantly produce lactase. And it's because their diet shifted, so they began consuming lots of dairy products. But a food intolerance is not an allergy. In the case of lactose, if you're lactose intolerant, you no longer produce lactase. So that means when you consume dairy products, the bacteria in your gut, especially E. coli, which loves lactose, will happily break it down and use it for energy. And in the process, those bacteria produce very unpleasant side effects like gas, bloating, and diarrhea because the bacteria are eating the lactose. And that's what causes your symptoms. But it's not an allergy. When it comes to the skin, localized inflammation leads to dermatitis. And so generally speaking, we're talking about localized anaphylactic reactions. But what happens when you have a systemic anaphylactic reaction? So in that case, the antigen, the allergen, which is usually injected somehow, so something like a bee sting, that triggers a massive release of chemical mediators, histamines, leukotrienes, prostaglandins. Your blood vessels dilate all throughout your body, and that causes your blood pressure to drop, and you can go into shock. You also get a lot of swelling, especially in the throat. And so your throat swells shut so you cannot breathe. So if you have a type 1 systemic anaphylactic reaction, you need epinephrine. Epinephrine can help to alleviate the swelling so you can breathe. But when you start having the reaction, you only have about 30 minutes, right, to get epinephrine, otherwise you will die. And so people who have severe allergies to things like bee stings or maybe certain spider bites, they carry EpiPens with them. So they carry epinephrine so that they can inject it if necessary. And if you do carry EpiPens, if you ever have to use one, you still must get to a hospital. And the reason why is the epinephrine does not last forever. It's going to wear off. And when the epinephrine wears off, it's possible for your anaphylaxis to return. So if you are in a situation where you're experiencing anaphylactic shock and you're able to inject yourself with epinephrine, you still must seek medical help. Any questions on that?
Can like a peanut allergy cause this too?
Oh yeah, peanut allergies will cause systemic anaphylaxis if you're severely allergic to peanuts. And so the problem with peanut allergies was bad advice. For decades, doctors told pregnant women not to eat peanuts or peanut butter while they're pregnant or nursing. And that was the exact wrong advice to give. It's the opposite. You should eat lots of peanut butter and peanuts while you're pregnant and nursing. But because so many women received this bad advice, over the past few decades, there's been an explosion in peanut allergies among children. And it's because their mothers avoided peanuts. And that was the wrong thing to do. But there are ways to, if someone has a severe peanut allergy, they can be desensitized to the point where if they do have an accidental exposure, they won't die. But, bad advice to avoid something, like peanuts, because that just made the problem worse. But peanut allergies can be very serious.
Wow.
So when it comes to these type 1 reactions, and in general to some of these hypersensitivities, you must have an initial exposure. And we call that first exposure the sensitizing dose. So the problem with the sensitizing dose is that you do not know that you've been sensitized. So let's say, for example, when you're a child, you got stung by a bee. And your immune system actually did make a response to it, but the bee venom, it's not around long enough, right? Because it takes your immune system 7 to 10 days. So by then, all the bee venom is gone, but your immune system may be ready for the next time. And so the next time you're stung by a bee, whether it's a year later or 10 years later or 50 years later, you have this massive overreaction by the immune system. So we call all subsequent exposures the shocking dose. And like I said, there's no way to know that you've been sensitized to something until you get that second exposure and have the overreaction. And you may be in a place where you cannot get medical help. And so you don't know you've been sensitized. You don't know you're going to have systemic anaphylaxis. So it can happen with things like bee stings. In the case of someone that used to work at Montgomery College, when I first started working here a long time ago, our microbiology course coordinator had become sensitized to iodine, but she didn't know it. And remember, with gram staining, you use iodine as part of the gram stain procedure. So she'd been using iodine for years, had no idea that she'd become sensitized to it until she had to go get a CT scan, and they injected her with the iodine for contrast, and she went into anaphylactic shock. So luckily they were able to administer epinephrine in time, but she had no idea she'd become sensitized to iodine. So it's possible to become sensitized to things you have no idea until you get that second exposure, and then the overreaction occurs. So hopefully you'll be in a situation where you can get medical help in time. So that can happen. Now, just to mention it, there have been documented cases of anaphylactic reactions occurring with no sensitizing dose. So there have been examples where someone has gone into anaphylactic shock with that initial first-time exposure. The exact mechanism is a little different, but it has been documented that the sensitizing dose is also the shocking dose In some cases. The overwhelming majority of the time, no. But it's possible, even if you had no prior exposure, you could have an anaphylactic reaction. So I just want to mention that because it has happened, so it is possible, but normally you must be sensitized first. And then all subsequent exposures, you have that overreaction. So what if you are allergic to tree pollen or grass pollen? And let's say springtime is really miserable because you're so sensitive. Well, the first thing they have to do is figure out, to what things you are allergic. And the way they do that is with a scratch test. And so they actually take the allergen, scratch it into your skin. If you are allergic, you will have a reaction, right? Your skin will swell up, you'll have a raised red bump, and the larger the bump, the more allergic you are to that particular allergen. And if it turns out you're allergic to a lot of different things, then it's worth becoming desensitized. And so allergy shots, as they're called, is a way to desensitize you. And what they actually do is inject you with the allergen. And they start off with very minute quantities, and then over a period of about 6 months, they build up to what's called your maintenance dose, the maximum concentration. And thereafter, you get a shot every month. And most people who start this have to do it for 3 to 5 years, at which point they may have become desensitized. Or if you're like me, no, right? But it does generally work well. And the way that it works is that by injecting you with the allergen, they're causing your immune system to produce IgG antibodies. IgGs are not attached to mast cells, so IgGs do not cause the release of histamines. But if you have a lot of IgGs, they can bind up the allergen so that it never binds to the IgE antibodies on the mast cells. And this can dramatically reduce the amount of histamines released and dramatically improve your symptoms. So they are very beneficial. In general, around 75% for things like pollen, but up to 97% for things like bee venom, right? It's for bee stings. So if let's say your job is that you raise bees, right? You have beehives. Well, obviously, if your risk of death is high from a bee sting, you need a way to desensitize yourself. So allergy shots can help. You just have to be willing to make the time to go get them every month. So those were type 1 anaphylactic reactions. Type 2 hypersensitivity reactions are cytotoxic. So these involve IgG or IgM antibodies and complement. Remember, the classical pathway to activate complement is when an antibody binds to something. And then the complement proteins will assemble and poke holes through cell membranes. The problem is that your cells are nothing but membrane. So if the antibodies stick to your cells, all the immune system sees is an antibody stuck to something, and that's a signal to attack and destroy. And so your immune system will attack your cells and tissues if antibodies are stuck there. Now, 2 examples of cytotoxic reactions are incompatible blood transfusions and the hemolytic disease of newborns. When it comes to blood transfusions, you should all be familiar with the ABO blood group. And the A and B antigens are actually carbohydrates attached to the surface of red blood cells. And the RH antigen, the rhesus antigen, is your positive or negative, right? So if you're positive, you have the RH antigen. If you're negative, you don't. So if you receive blood that has these antigens on the surface and your blood does not have these antigens, your immune system sees those blood cells as foreign and will attack and destroy them. So if you're type O, for example, and you're transfused with type A blood, all that blood is foreign, right? Your immune system is going to see the A antigen and will attack those red blood cells and destroy them. And that's going to clog your capillaries and lead to organ failure and death. So when they type blood, there is a reason why, because your immune system will attack blood cells that have antigens that you do not have. And so remember, type O has none of the antigens, and in particular, if you're type O negative, you have none of the antigens. So your blood is highly desirable because O negative blood can be given to anyone because there's no antigens on it. So everyone can receive O negative blood. But O negative people can only receive O negative blood and nothing else. And type O does tend to be the most common blood type, or at least it belongs to the largest percentage of people in most populations. But when they ask for blood donations, they especially want type O blood because that can be given to anyone, especially O negative blood. But O negative can only receive O negative. If you're type AB positive, the rarest type, you can take any blood, right? Because you have all the antigens, so all blood works for you. When it comes to hemolytic disease of newborns, I still like the old name erythroblastosis fetalis, but this involves the Rh antigen, so the rhesus antigen. So dad is Rh positive, mom is Rh negative, but the fetus is Rh positive. Now, when it comes to the fetus, in general, half of all the antigens in that fetus are foreign anyway, right? And so mom's immune system does an excellent job tolerating that fetus because half the fetus is foreign, and mom's immune system ignores it. But what can happen if there are complications during a pregnancy, or especially during birth, the fetal blood can come in contact with mom's blood. So mom's immune system can see the Rh antigen. If this happens at delivery, there's no problem for that baby because it's already been delivered. It's gone. But mom's immune system will make antibodies against the Rh antigen. So in future pregnancies, if that fetus is Rh positive, mom's IgG antibodies will cross the placenta, attack the fetal red blood cells, and kill that fetus. So what that happens, if mom is Rh negative, and dad is Rh positive, and the fetus is Rh positive, and somewhere along the lines, mom's blood came in contact with the fetal blood. And so her immune system made those antibodies. Now, if they know who daddy is, or if they do an amniocentesis and they know the blood type of the fetus, and this situation exists, then what they can do is give mom a preparation of antibodies called RhoGAM, and that's sort of like the brand name, RhoGAM. And so these are antibodies against the Rh antigen. So the idea is you inject mom with RhoGAM, and those antibodies will bind up any Rh antigen that's present, so her immune system never sees it and never makes antibodies against the Rh antigen. So but this is a case where mom's immune system is attacking the fetus, is attacking the fetal red blood cells. And it's not something that we want to happen. But this is a cause of stillbirth in women if they are Rh negative and their partners are Rh positive and their fetuses are Rh positive. So this can and does happen. Any questions on blood transfusions or hemolytic disease in newborns?
Is it common? Is this a common thing?
I don't know if it's common, but it is something that they will test for, especially if mom is Rh negative. And then they want to see what's dad's blood type. And if they don't know who dad is or if he's not around, and then they can do an amniocentesis and figure out the blood type of the fetus. And if that's the case, then they can administer the RhoGAM. But I know this has happened. It happened with my aunt who had many miscarriages before she finally had 2 girls that were Rh negative. So it does happen. Type 3 reactions, also known as immune complex reactions, this involves the mechanism of precipitation. So when antibodies bind to soluble antigens, they become insoluble and they precipitate, they become solid. The problem is these immune complexes can get stuck to your tissues. Your immune system sees antibodies stuck to your tissues, so the immune system starts to attack your tissues. But this is an inappropriate response. It should not be happening. It's just, these complexes happen to get stuck, and then your immune system attacks. But it should not be happening. Often, things like the kidneys, joints, and lungs are involved in this. An example is glomerulonephritis, where the complexes get stuck in the kidneys, and the immune system starts to attack the kidneys. But this should not be happening. The complexes are just an accident where the antibody happens to get stuck to your tissues, but your immune system knows to attack anything that has an antibody stuck to it. And if that's your tissues, then that's what the immune system attacks. When it comes to type 4 reactions, these are cell-mediated, in particular T-cell mediated. And because they're mediated by T cells, the reaction is a bit slower. It may take 24 to 48 hours after contact with the antigen for you to make the response. Normally, there's a sensitizing dose and then a shocking dose, right, for all subsequent exposures. When it comes to the skin, when your skin comes in contact with the antigen, it leads to inflammation and destruction of the epithelial cells, so that causes blisters. We mentioned urushiol oils from poison ivy before. So let's say you're out playing in the woods and you come in contact with poison ivy and don't know it, so you got it on your hands, and then you touch other parts of your body, so you're spreading the urushiol oils, but you don't break out for a day or two. And then it may look like your poison ivy is spreading, even though it's not, because you've already spread the oils. It's just taking some time, right, for those other parts of your body to experience the reaction and the blisters. So if you're out in the woods, it's important, if you came in contact with certain plants, to wash your hands and arms thoroughly. Any other parts of your body that you may have touched or come in contact with the poison ivy, because if you can get the urushiol oils off fast enough, that will stop the reaction from occurring. Some people are allergic to poison ivy, others are not. When it comes to cosmetics, certain cosmetics can cause allergies too, right? Dermatitis. Soaps and detergents, especially the chemicals they use for fragrance, those are often very allergenic. Drugs like penicillin, right, can cause these kinds of allergies. Heavy metals like nickel. If you buy really cheap jewelry, you may get a rash where that jewelry comes in contact with your skin if you're allergic to some of the heavy metals used to make that cheap jewelry. So it's not sterling silver or gold or anything. And iodine, as I mentioned, iodine is something that some people can become allergic to. Most of these things are haptens. Remember, the haptens are antigen wannabes, so they need to come in contact or stick to a tissue protein in order to become an antigen. But again, these kinds of things are not dangerous, right? So poison ivy is not dangerous, right? Cosmetics and soaps are not dangerous, but your immune system is overreacting to them as if they are dangerous. Here we see an example of contact dermatitis, most likely a latex allergy and wearing latex gloves. Remember, latex is a natural product, right? So the latex rubber is a natural product, but it often has contaminating proteins mixed in with it, and it's the proteins that are the allergens. And so some people are very allergic to latex. Fortunately, we have a lot of alternatives today, like PVC gloves or nitrile gloves. And so we always tell our students not to buy latex gloves, right, just in case, because even if you're not allergic right then, it's possible that you become allergic in the future if you become sensitized to it. And the question here, which hypersensitivities are humoral? So that means which ones are mediated by antibodies. We saw that type 1 reactions are mediated by IgE antibodies. Type 2 reactions are mediated by IgG and IgM antibodies. Type 3 reactions are immune complex reactions involving antibodies. So types 1, 2, and 3. And type 4 is T-cell mediated. So those are the major types of hypersensitivity reactions. When it comes to autoimmune diseases, very often these suppressed self-recognizing T and B cells get reactivated. And so in that case, they can lead to the production of autoantibodies, antibodies against the self. And this leads to autoimmune diseases, a loss of self-tolerance. So all the different types of hypersensitivity reactions can cause various autoimmune diseases. An example of a type 1 reaction is something like Lyme disease. So Lyme disease is caused by the bacteria Borrelia burgdorferi, which has a protein on its cell wall. This protein is actually similar to a human leukocyte antigen, an LA, that some people have. So what that means is if those people get infected with the Lyme bacteria and make antibodies, those antibodies are cross-reactive and can attack various tissues in their body because of that similar HLA antigen. And a common consequence of Lyme disease infection is autoimmune arthritis. So the immune system starts to attack the joints because the immune system is trying to attack the bacteria, which may no longer be there, but the antibodies against that antigen, which is similar to one that some people have. And so the antibodies are, binding, right, to certain tissues and causing an autoimmune disease. When it comes to the rest of these hypersensitivities, including type 5, the stimulatory hypersensitivity, we're going to look at some specific examples of those. And we'll see how the type 5 is similar and different from our type 2 cytotoxic reaction. So an example of a type 2 reaction is Graves' disease. And the thing about Graves' disease, this is one example where it is a stimulatory type of reaction rather than true cytotoxicity. So it involves the thyroid gland. And when antibodies bind to the thyroid, what happens is the thyroid gets stimulated to produce excess thyroid hormone. And that causes a wide variety of symptoms. But the thyroid is not really being attacked by the immune system, but it is causing the thyroid to be overstimulated. And this condition is treatable. But this is an example of what we would call cytotoxic, but the British would call stimulatory, because the thyroid is truly being stimulated rather than being attacked. When it comes to type 3 immune complex reactions, we have two examples, lupus and rheumatoid arthritis. So you may have heard of lupus. There's been a lot more coverage of lupus in recent years, but this is an autoimmune disease where those individuals produce autoantibodies. So they produce antibodies against a wide variety of antigens in their bodies. And the thing about lupus is it can manifest itself very differently. In some people, lupus is a very mild condition, so mild they will never suffer any serious problems. And in other people, it kills them. The thing about lupus is that you have periodic flares, right? So your lupus will flare up, and then it'll be brought down under control, and then flare up, and then be brought under control. So if you have the severe form, you will have flare-ups. And we had, many years ago, we had an administrative aide in the math department, and she had lupus. But to look at her, talk to her, you would never know there was anything wrong with her, because every once in a while she'd have a flare-up, but most of the time her lupus was under control. But when she was about 52, she had a flare, and they couldn't bring it under control, and she died. So lupus can be very mild, it can be very serious. It just depends on the individual. When it comes to rheumatoid arthritis, here the immune complexes get stuck to joints. And so the immune system begins to attack the joints and causes a lot of inflammation and a lot of arthritis. So it's a very painful condition. In severe cases, the joints have to be surgically removed and replaced with artificial joints. But these reactions should not be happening. Right? They are inappropriate responses where the immune system is causing damage or death to the host. When it comes to type 4, a good example of a T-cell mediated reaction is type 1 diabetes or insulin-dependent diabetes. In these individuals, their immune system has attacked and destroyed the pancreatic cells that produce insulin. So type 1 diabetics cannot produce insulin, and that's why they're completely dependent on insulin. And we typically see this in juveniles, right? So it's juvenile diabetes, but it is becoming more prevalent in adults. So for some reason, in the adults, they're now getting this autoimmune condition where their T cells are starting to attack the pancreas, and they're not sure why. But one of the triggers is often an infection, and usually a viral infection. And so as the immune system is fighting the virus, somehow it becomes sensitized to the pancreas and to those cells producing insulin. I had a student a few years ago, and that's exactly what happened to him. When he was a teenager, he got some miscellaneous viral infection. He recovered in a couple of weeks, and then a few weeks after that, he almost went into a coma because his blood glucose shot up really high, and no one had any idea, right, that he no longer could produce insulin. So luckily, he was able to get to a hospital and was successfully treated, and now wears an insulin pump, so he can maintain normal blood glucose. But it was the result of an infection that he had when he was a kid. But, most of the time, this does not happen, but in some people it does. But this is an example of an autoimmune disease, a type 4 reaction, right? Type 1 diabetes. When it comes to organ transplants, the idea is that you're receiving foreign tissue, and the immune system sees that foreign tissue and says, oh, no, and the immune system is going to use everything it has to attack and destroy that foreign tissue, right? T cells, B cells, antibodies, complement killer cells, everything, right, to destroy that foreign tissue. But of course, we don't want that to happen. Now, there are certain places in your body that are privileged. We have privileged sites and privileged tissues. And the difference is a privileged site, the immune system does not go there. And the reason why we know that when the immune system is fighting, it does incidental damage, right, to the host. In places like the brain, that damage cannot be repaired. It's permanent. In the cornea, you damage that cornea, your body cannot repair that damage. So we do not want the immune system going to certain places at all, because any fight that goes on in those places will cause permanent damage. When it comes to the testes in males, those are privileged sites. The immune system does not go there. But ovaries in females are not privileged, right? The immune system goes there. So why do you think the testes are privileged sites while the ovaries are not? What makes us males so privileged? Why are we so special? Any ideas why the immune system does not go into the testes? Any ideas? So Bryant and Craig, do you know why you're privileged? No guesses? What do the testes produce? Sperm. When do they start producing sperm? So when do males start producing sperm?
At puberty?
At puberty, right? Around age 11, 12, 13. When does the immune system learn to recognize self from non-self? When does the immune system learn what is self? During fetal development, right? So when males start producing sperm, the immune system never seen them before. So sperm are foreign to the immune system. And if the immune system were to see those sperm, the immune system would attack and destroy them. And that's actually one of the causes of male infertility. Somewhere along the line, there was an accident or injury which caused the immune system to be able to see those sperm cells. And now the immune system attacks and destroys them. But when it comes to eggs in the ovary, they actually form during fetal development and go through meiosis 1 and then stop and sit and wait. But the eggs are always there, so the immune system has seen them. But sperm, the immune system has never seen them before. And if the immune system does, the immune system will attack and destroy. So that's why you do not want the immune system going into the testes or the brain or the cornea, because it will do damage there, and that damage cannot be repaired. A privileged tissue, on the other hand, the immune system sees it but doesn't care. But the immune system ignores a privileged tissue. So things like heart valves. You can transplant heart valves from one person to another, no problem. The immune system does not care. Same with the Achilles tendon, right? That tendon on the back of your foot along the heel. If you rip that apart, doctors can't just sew it back together, but they can replace the tendon from one that came from a cadaver, a dead person. Usually they freeze it first, as there might be a few cells left there from the other person. But the Achilles tendon is just protein. It's all the same kind of protein. And so the immune system doesn't care about it. And then I already mentioned the fetus, a remarkable example of immune tolerance by mom, right? Because half of the genes creating that fetus are foreign. So half of the antigens in that fetus are foreign. And mom's immune system ignores it. And the fetus really is basically just a parasite, right? The fetus steals food from mom, right? It steals nutrients, it steals oxygen. It uses mom as a trash can for waste disposal. I mean, there's no benefit, right, to that fetus, to mom. It's really just a parasite. And yet her immune system ignores it, all right? You have this 50% foreign object growing inside of you, and mom's immune system is just like, yeah, whatever, right? So, and if you're lucky, it's only a parasite for 9 months, and then 18 years after that, right? So let's say you need a transplant. That's called a graft. And so an autograft, not to be confused with an autograph, it's not a signature. An autograft is simply moving a tissue from one part of your body to another. So that's not a problem because it's all your tissue. But what if you need a kidney transplant? Well, if you have an identical twin, that would be an isograft, and that would be the best possible match. Although even identical twins are not 100% antigenically identical, but they're the best possible match. But how many of us have identical twins, right? Not many. So we have to rely on other people, and those are called allografts. So a tissue from other people. And obviously that's foreign tissue, right? So they type and match very carefully. The xenografts are things that I really think we should stay away from. And those are transplants from other animals, from other species. The concern that I have is that we don't know what kinds of viruses and things might be lurking inside the animal and what might happen when that gets inside of us. So I think that we should stay away from xenografts. Now the problem with allografts and even with isografts is that the host can and will reject the graft. So normally the host rejects the graft, but there is a case where the graft rejects the host. That happens with bone marrow transplants. Because remember, all of your blood cells, including your immune cells, are produced in the bone marrow. So when you receive a bone marrow transplant, you're putting someone else's immune system into your body. So your body is foreign to that immune system. And so the graft can reject the host. So that transplanted bone marrow can start to attack you. So that's why bone marrow must be very, very carefully typed and matched. But even still, it's not a perfect match. So as I said, rejection is the main problem because your immune system is going to want to reject that foreign tissue. And your immune system is going to use everything it has to try to destroy that tissue. So how can you tolerate the transplant? Well, you must take immunosuppressive drugs, like cyclosporin is one example, but there are many others. But you must take these drugs which suppress part of your immune system so you can tolerate the graft. Otherwise, your immune system will quickly reject, right, that graft. But think about what you're doing. You're suppressing your immune system. So that makes you more susceptible to infections and to cancers because you're suppressing part of your immune system. But that's the trade-off. if you need a transplant to survive, you put yourself at higher risk for infections and cancer. So you must be more vigilant, right? More regular checkups, and be very careful when it comes to infectious diseases. So, but you don't have any other choice. One day we will be able to grow whatever organs you need, right, for replacement. But we're not there yet, but they are making progress. But ideally, one day we will be able to grow whatever organs you need for transplants. But right now, we still have to rely on organ donors. So any questions or comments?
Yeah, I've always been curious. How do they match someone? Like, they have similar
Well,
DNA.
well, obviously they look at blood type, but they also look at other various antigens to see. But the best possible match, again, is from an identical twin. But otherwise, they give you what's reasonably available, and then they suppress your immune system so you tolerate that transplant. But it's an ongoing, battle because a faculty member has a kidney transplant, but that's the second one that he got because the first one he just rejected very badly. And so he did get a second one, which he tolerated much better. One of my former students, when he was like 13 years old, again, he suffered from some infection, and then he got an autoimmune reaction, which destroyed his liver and needed a liver transplant at like age 13. So his parents are always extremely worried about a cold and flu season because he's much more susceptible. But he goes in for regular testing, and they take biopsies of his liver to make sure that the liver is still okay and not being attacked, right, by his immune system. But at some point, he is going to need another liver transplant. So when he got, it was the only one available, and it wasn't the best possible match or the best possible source. And it was much older than he was. So he's going to need another transplant probably in a few more years. So, but that's the trade-off is you must have these organs to survive, but you must also suppress your immune response to tolerate that transplant. And that makes you more susceptible, right, to infections and cancers. So,
Wouldn't someone's parents be the best option?
well, in the case of a liver transplant, here's the problem. If you receive a whole liver, all the connections, all the blood vessels, right, all the arteries and veins, it's much easier to connect everything. But if you take a piece of a liver, like from the parent, then you have to completely reroute all the plumbing. And once you do that, you can never receive a whole liver transplant. You can only get pieces. And so that's why if you get a whole liver, they can easily reconnect all the blood vessels and everything. But if it's a piece of a liver, they have to go in and reroute and change all the connections. And that means you can never receive a whole liver transplant. So,
Oh, wow.
yeah, so it's best if they can find a donor, right, to donate an entire liver. But if there's no other option, then they will take, one of the lobes, because your liver has multiple lobes, like 3 lobes, and they can take a piece of that and they can transplant it, but they really have to work at, reconnecting and reconstructing blood vessels to make sure that everything is, is receiving the proper blood flow, and that messes everything up. So, liver are not the best things to have to get in the first place, but a whole liver is much better than a piece of one. Yeah, someone else have a question? No. Okay, so let's start lab around 20 after. All right.
Thank you.
So I'll see